Have you ever wondered why the structure of carbohydrates are look like this, this, and how do we represent the structure? And this structure is actually a proper representation of carbohydrate? Well, let's find out in this video. This is structure of carbohydrates based on the three types of representation by Venkadesh, and welcome to Food Catalyst. So carbohydrate is actually an organic compound. Okay, carbohydrate is an organic compound. So why do we say this is an organic compound? If the carbon is present in a compound, then we call it as an organic compound. For example, in the carbohydrate, three main components are present. One is carbon, hydrogen, and oxygen. Okay, it's carbon, hydrogen, and oxygen. Since carbon is present, it's an organic compound. Okay, wherever carbon is present, then that compound is called as organic compound. If carbon is not present, it's called as inorganic compound. It's called as inorganic compound. Okay, so apart from that, carbohydrate consists of carbon, hydrogen, and oxygen. And we need to know one thing, always hydrogen and oxygen in the ratio of 2:1. What do you mean by that? It means the hydrogen ratio will be 2 times higher than the oxygen. So it will be always in the state of H2O. Okay. What is H2O? It's water. Right? So C plus H2O. Okay. C plus H2O. C means carbon. And what is H2O? We will call this as a water or hydrate. Water or hydrate. So combining these two, we can get carbon hydrate. So ultimately we call this as a carbohydrate. Carbohydrate. Okay, so now we understand what is a carbohydrate. But in a much complex format, we have to name this as polyhydroxy. This is very important. Polyhydroxyaldehyde or polyhydroxy ketone. This aldehyde and ketone are the functional groups. And there are some derivatives from these two, polyhydroxyaldehyde and ketone. So that's the carbohydrate. And there is one more thing, CHO. See, CHO means basically the term CHO here it's actually called as aldehyde, and C double bond O is ketone. We already know that, all the chemistry people. Okay, see, this term we simply call that as a saccharide. This functional term we call this as a saccharide, means sugar. If a component has only one saccharide, it's mono. If it has two, then it's disaccharide, then trisaccharide, and so on. Let's take an example of glucose. Okay, so glucose is actually C6H12O6. So glucose has only one CHO. So that means it is a monosaccharide. Okay. So these are the important information you need to know before we get into the topic of structure of carbohydrates. Okay. So what is a structure of carbohydrates based on three types of representation? See, we can divide the structure into many types, right? We can even divide the carbohydrates into functional group, as I told you, aldehyde sugar or ketone sugar, and we can divide into number of carbon atoms. If that is a 3 carbon atoms, it is triose, tetrose, pentose, and hexose, and so on. But here we are going to see about the representation of carbohydrate. Okay, see, basically the structure of carbohydrates is based on the three types of representation. So what are the three types? The first type is Fischer projection. So Fischer projection is the expansion of molecule. Okay, it's nothing but an expansion of molecule. I'll explain this in detail, but here we need to understand two rules. So the first rule is carbon. So the carbon will be bonded with 4 different atoms. So 4. So carbon has 4 bond which is left. This is based on the valence reaction. You don't need to know understand why. But here you can see each carbon will be bonding with 1, 2, 3 and 4. Right? So each carbon is actually bonding with 4 different atoms. So this is what we need to understand here. So carbon has 4 bond. And what about the other 2? Oxygen. Oxygen has, here you can see, simply 2 bond. So oxygen only have 2. Then hydrogen. Hydrogen has only 1. So the first rule is to understand how much of bonding will happen in an each atom. So for carbon it's 4. I'll write it here. For carbon it's 4. For hydrogen it is 1. And for oxygen it is 2. So that is the first thing. Then we have to expand the molecule. So here I have taken the example of glucose. So I have taken an example of glucose and I have expanded it. And as you can see, the CHO, actually it simply sums up to be the CHO. Means it has only one CHO in the whole molecule. And that's why this glucose molecule is monosaccharide. That we have already discussed. And this glucose molecule has how many carbons? It has present 6 carbon. Okay, we'll discuss that detail later. So this is the first representation of carbohydrate. Then here comes the Haworth projection. Then conformational formula. So let's go deep into each and every projection and why it is important and how we have come up with this projection. Let's explain this in detail. First is Fischer projection. Fischer projection is nothing but the position of hydroxyl group. So what is a hydroxyl group? Hydroxyl group means OH group. Okay, simply it means OH group. So let's take the same example of glucose here. See, as you can see, I have hidden the carbon here. So in this way, instead of writing this in this big reaction, like as you can see, by simply hiding the C, I can get a shorter and simpler reaction. So this is a one easy way to represent the Fischer projection. Okay, so let's see this molecule in detail. See, how I have hidden is wherever there is a bond in the straight line, that means C is involved. Okay, so if there is a bond in the straight line, that means C is involved. That's how we need to understand the carbon. So here, how many carbons are present? Here, 1, 2, 3, 4, 5, and 6. Because we have 1 here, 2 here, 3 here, 4 here, 5 here, and 6 here. So 6 carbons is present in the glucose, and that's why it's a hexose, and that's why we call this as a monosaccharide. I already told you that reasons. Now let's talk about the other components. See, Fischer projection, let's start with the simplest monosaccharide. What is a simplest monosaccharide? It's triose. Okay, the 3 carbon compound. 3 carbon compound. This is how the simplest we can get. The 3 carbon compound of Fischer projection of the monosaccharide is the simplest form of monosaccharide. What is the simplest form of monosaccharide? It is the 3 carbon compound triose. And in the 3 carbon, we have the glyceraldehyde. Okay, which means there is an aldehyde in the top of the molecule. Okay, so here there is only 3 carbon, and as I can see, the second carbon is hidden. Okay, and then tetrose. So tetrose means what? 4 carbon is present. 1, 2, 3, 4. And same, it's pentose, so 5 carbon is present. I have given the example of ribose. And then next is glucose, which is 6 carbon. And same, I have fructose, which also has 6 carbon. Let's see. 1 here, 1 here, 1 here, 1 here, 1 here, and finally 1 here. So here also 6 carbon. See, as I told you, each and every bond is actually representing a C. So here is also a C is present. So you need to understand that. Okay. Okay, this is C double bond O. This is the functional group here. Okay. Okay. So see here, as you can see, this has a functional group of CHO. So that we call that as a aldehyde, right? Aldehyde. So it is called as aldohexose. So here ketone is present. So it is ketohexose. Okay. Basically, hexose means 6 carbon. We already know that. By implementing the functional group, we will say that as an aldohexose and ketohexose. So if they ask in a simpler manner, what is a Fischer projection? Which means the position of hydroxyl group. See, in all these structures, the one thing is clear about where this OH is placed, right? But let's take an example of glucose and understand the situation. See, there are many components like this are there, but how do we understand the glucose? See, to understand a 6 carbon glucose, we need to understand where the OH is placed. So here we can see in the third carbon, OH is placed in the left side. That means that is a glucose. So this is how we need to come up with the idea and strategy so that the position of hydroxyl group will ultimately determine the structure of a component. Okay, so this is the actual reason behind the Fischer projection. But now you understand the Fischer projection, let's move on to the further. See, Fischer projection, we can see that hydroxyl groups are in the left or right, right? So the above format is conventionally true. It's true. But imagine writing a long reaction with this bit interpretation. What will happen? If I writing for every reaction, if I'm writing like this, then what will happen? So that's why Fischer projections cannot be written in the straight equation. We need a cyclic reaction to make it use in the bigger interpretation. So how do we make it cyclic? See, the Fischer projection can be written in the cyclic hemiacetal or chemiketal form. So don't worry about what is hemiacetal or chemiketal. I will explain this in detail. But first of all, why cyclic formation, right? Why we need to choose a cyclic formation first of all? See, take an example of these three: aldo, tetros, pentose, and hexose. In solution, they will undergo cyclization. Okay, I mean, while in the condition of solution, it will undergo a cyclization reaction. And in the cyclization reaction, it will form two types of cyclic components. That cyclic component, if it from aldose, means it hemiacetal. If it is ketose, then it is hemiketal. Okay, so how do the cyclic compounds first of all form? Let's take an example. So I have taken the same example, glucose here, as you can see, which is an aldohexose. Why I am telling this as an aldohexose? I'm explaining because it has 6 carbon compound and it has an aldehyde compound. Okay, so here is, as you can see, this has one CHO group and 5 hydroxyl group, either in the left or right. I have marked down all the hydroxyl group and the aldehyde group. Now let's take an example of the cyclization reaction. See, the cyclization reaction always happen. This is important. It's always happen with the functional group. Okay, with the functional group plus the fifth carbon group. So this cyclization reaction is happening within the component. For example, this cyclic reaction is happen within the glucose. I am not adding one glucose to another glucose. So if it is within the same molecule, then we call that as an intramolecular reaction, which means within the molecule. If I am adding glucose plus fructose, then that means intermolecular reaction, right? You need to know the difference between intra and inter. Okay, so this is intra, we already have seen, and as I told you, the functional group is reacting with the fifth carbon. But how it is reacting is very important. As you can see, the H is attracted to the O, which means the bond in O will break and the H will come and join the O. But what happens when the H will join the O? There is one bond left for the carbon. See, as I told you before, there is 4 bond needed to carbon to survive. If there is one bond left, then what that carbon will do? The carbon will try to attract the oxygen, because here, as you can see, if the OH has been went from this area to this area, there is a bond left in oxygen too. So the C will come and bond with the O and the H will go from here to the O. So let's take the example here. see, I have clearly mentioned this H has been, the H here has went here. Okay, the H here has went here. So the H's has been like transferred here from there. And what about the O? See, the O has one bond left and carbon has one bond left. So ultimately the C is connected with the O, forming a cyclic structure. Now it is a cycle. As you can see, it is enclosed. So this cyclic structure is formed because of the solution formation. Okay, the solution formation will ultimately give the cyclization reaction. Okay, so during cyclization, intermolecular reaction happens between the CHO and C5OH, right? C1CHO and C5OH, and linking the carbonyl atom with the other carbon atom through COC linkage. See, here, as you can see, this carbon is ultimately linked with the first carbon through a oxygen molecule. This is called a COC linkage. First carbon and the fifth carbon has been linked through an oxygen. Okay, this produced a closed ring that we call that a ring as a pyranose ring or cyclic hemiacetal. We have already seen it is a cyclic hemiacetal. But why pyranose ring? Let's take that example. I'll explain the pyranose ring later, but let's take the same example. See, the glucose has been transferred, the H has been transferred to the first carbon, and the C has joined the bond with the O and make a COC linkage. So because of the first carbon has a cyclic link with the fifth carbon, it made a COC linkage. Now we know that. But see, C1 becomes asymmetric. So earlier it was symmetric. Until the oxygen was there, it is symmetric. Now it has been linked. Now we can see there is a different component in the each side of the carbon, in the each side of the first carbon. So this different types of, this asymmetric structure result in two types. Because see, if suppose the OH is present in this side or this side, so there is a two possibility. I can write this reaction in the two possible way. So that's why we need to know that there is a two possible reaction. So this asymmetric carbons will have different molecules. So see, if this is very important, if the OH is present in the right side of the component, it's called as alpha formation. If the OH is present in the left side, as you can see, it is beta formation. So whether this is an alpha or beta formation, that's how we need to know if this is alpha glucose or beta glucose. Okay, so let's write the reaction accordingly. See here, as you can see, the OH is present in the right side, and here as you can see, the OH is present in the left side and right side, right? So if it is in right side, it is alpha. If it is left side, it is beta. So basically this differentiation is called as anomers. So what is an anomer? Anomer differs only in the configuration of C1 and the C1 compound, which ultimately components as an anomeric carbon. And see, this C1 has to be in an asymmetric format, because that's why we get alpha and beta. Otherwise we will get only one component. Why we get the two component, right? It has to be in a two different state, alpha and beta. Okay, so it should be in a asymmetric format. That's what I'm telling you, asymmetric format. And this C1 carbon is called as anomeric carbon. So what is an anomer? Anomer means the configuration will change around the C1. So based on the anomers, we'll say whether there is an alpha or beta. And the carbon which determines that, the first carbon, it's called the anomeric carbon. So understanding what carbon is anomeric carbon is really important to determine whether it is alpha or beta. Okay, so let's move on. Now, okay, now let's talk about Haworth cyclic structure. First of all, this Fischer projection does not accurately describe the two shape of cyclic. See, I will tell you this as a cyclic because it has a closed bond, but this is you have seen the other cyclic structure, right? This is actually doesn't look like a cyclic structure. So to represent the true shape of cyclic structure of hemiacetal or hemiketal, Haworth new formulation came up with the idea called flat planar hexagons. What is a flat planar hexagons? I'll let you know. First of all, this Haworth new formulation, the Haworth's new formulation is called as Haworth cyclic structure. Okay, so let's take an example of the glucose here. See, I have taken the same example of glucose here. So glucose is a 6 carbon compound here, as you can see. It's a 6 carbon compound. So I have mentioned all the 6 carbon and 1, 2, 3, 4, 5, 6. And where is the aldehyde present? Aldehyde is present in the first carbon, so which is the functional group, and fifth carbon. And fifth carbon is obviously fifth carbon is also important. Why? Because the first carbon will link with the fifth carbon to make the hemiacetal. I have already discussed that, right? So here, as you can see, I have bended the structure in such a way that you can easily understand how it will link. See, this is the same aldehyde group, and so this is the OH group. See, as you can see, this bend will try to happen. So instead of making it a line or something, here we are bending the whole structure from first carbon to sixth carbon, so that this O will bond with H, and this this H will come and bond with the O, and this O will join with the C. Okay, I am telling you again. 1 second. See, this OH and the CHO will try to bond. That is the hemiacetal or hemiketal. We have already discussed that first carbon linked with the fifth carbon. Sorry. So how it will link? The H will come and join with the O, and this O will directly bond with the C. So, but here it attaining a cyclic structure. As you can see here, it is attaining a structure. As I told you, there is two alpha and beta, right? So here alpha will be, see, if it is representing in a cyclic manner, as you can see, the same cyclic manner, if the OH is present in the downside of the sixth carbon, see, this is an easy way to understand. If the OH is present downside in the sixth carbon, it is an opposite side. If the OH is an opposite side of the CH2OH, then it is alpha. If the OH is present in the same side, then it is beta. Okay, so according to the where the OH is present, corresponding to the CH2OH, we can say whether it is alpha or whether it is beta. Okay, this is an easy way to understand. So now you have understood it. Let's try to name the two different structures. The first structure. So as you already know the first word of the structure, which is alpha. Okay, and the second word is actually D. I will explain the D later in another video, why it is called as D or L. So let's mention D. And this is a glucose, right? Ultimately, I have converted the glucose into this structure, so it is gluco. And I told you before, it will make a pyranose, so it is glucopyranose. And what about the structure B? It is beta D glucopyranose. The structure is beta D-glucopyranose, which is because it is in the same side of CH2OH. If it is alpha, it is in the opposite side of CH2OH. So here you can see, if it is in the right side, it is alpha, left side it is beta. Since it is a cyclic structure, we are understanding through CH2OH whether it is in the opposite side or same side. Okay, so let's take a few examples. Let's understand few other points. The 6-membered ring shown for alpha and beta D-glucose is called a pyrene ring, right? I told you it's pyrene ring. The pyrene name is a heterocyclic compound consists of 5 carbon and 1 oxygen atom. If a ring contains 5 carbon, 1, 2, 3, 4, 5 and 1 oxygen atom. Okay, 1 means 5 carbon and 1 oxygen means it is a pyrene ring. Okay, to mention that ring as a pyrene rings, as you can see here, ultimately this is a 5 carbon. See, you cannot consider CH2OH now, right now. In a closed cycle, we have a 5 carbon and 1 oxygen. So this is ultimately called as a pyrene ring. Okay, so any CHO containing a 6-membered ring. So see here, they are mentioning 6-membered ring including the 5 carbon and 1 oxygen atom. Okay, so any CHO containing 6-membered ring is called as pyranose. Os, we have to add the Os because it is glucose, and CHO will be there. Os we have to add, right? So it is pyranose. So I have taken since like we have understood the glucose, let's take an example of fructose. So same, I have divided this into 6 carbon and we have to bend the carbon, and now this here is an important part. As I told you, the functional group will be linked, right? So this is the functional group, and here it will link with the 5th carbon. So here also same, it will link with the 5th carbon, and here is a functional group. So it's here 2nd carbon linking with the 5th carbon. So what happens, the 2nd carbon is linking with the 5th carbon, it will create the same, the OH will be bonding and C will be linking with this, C will link with the oxygen, and the another COC linkage will be formed, and after forming the COC linkage, the structure will look like this. Okay, so here see, obviously this OH and CHOOH will be replaced here and there. I have already explained you, if the OH is in the opposite side of opposite side of this CH2OH, then it is called as alpha. If it is in the same side, if suppose the OH is present in the top corner here, then I would have called that as a beta, but here it is an alpha. Okay, here it's called an alpha. So how do we name this structure then? So it's simple. First of all, we'll start with an alpha, obviously, then D, then it is a fructose. So fructose, here the structure is called as furan. So it is actually furanose. Okay, so this is a cyclic structure. Okay, so this is furanose. So let's study some important facts about furanose also. The carbohydrate containing 5-membered ring. See, this is not 6-membered because it consists of how many carbon? 1, 2, 3, 4. 4 carbon and 1 oxygen. So 4 plus 1, 5. 5-membered ring. Okay, so because furan is made up of heterocyclic component of 4 carbon and 1 oxygen. This is the structure of furan. As you can see, the same structure is here. Furan structure is here. So this is the same structure of furan ring. Okay, so see, next we are going to talk about the conformational formula. See, Haworth structure is not planar. Even though he said that is a planar, but it is not planar. It does not represent the correct formation of monosaccharide. So then what should we do? That's why we make the 2 type of conformational formulas. One is a chair conformer, then boat. Somehow they thought this structure, like this structure is look like a chair for them, and this actually they thought that this is look like a boat, and that boat is also fine, but I don't know why this is chair. But this is a chair conformer, chair structure, chair conformer. This is boat conformer. So basically they are representing the same cyclic component here, but they are representing in such a way that it is look like a boat, or this is look like a chair. But why do we represent this in a different manner? I will explain. See, this is a planar structure which we have seen earlier. As you can see, this hutch and that cut is actually interluding with one another, right? Then how do we understand the structure? Several angle strain is there, eclipse strain is there, and small steric strain is there. So because of this strain, we cannot look carefully into the planar structure, so that we cannot like fully utilize it. So that's why they came up with the boat structure, so that each and every hutch will not interlude with one another. But as you can see, even though it is not interluding, this angle strain is there. Like how do we even come up with this angle, right? And steric overcrowding of the 2 hydrogens. See, this overcrowding is there. It's not interluding like before. Like the planar angle, the boat conformation is not interluding, but still it's keeping a crowded place. Next comes the twist boat conformation. It's even more terrific. Here it's not overloading, but the strain is there in each and every angle. We cannot simply draw this angle. Finally, they have come up with this structure, which is much more easier to write, and much more gap formation is there between one hutch and another hutch. So no angle strain, no eclipse strain, small steric strain. So if someone asks you which is the best way to represent a glucose, then chain conformation is the best way. Okay, if they ask you more stable, then chain conformation. Why? Because when we talk about the steric relationship, like the mentioning the compound steric relationship, this chair conformation is the most stable one compared to all other three, right? As I have explained before. So that's it. That's how they came up with the best structure of carbohydrate and how to represent the carbohydrate. Okay, so let's answer some of the important MCQs which is asked in the previous year papers and then other competitive examinations. Okay, so follow our Telegram channel. So let's move on to the MCQs. So which of the following Fischer projection formula is same as the D-glyceraldehyde? So first of all, we need to know that glyceraldehyde has a 3 carbon compound, 1, 2 and 3, right? And it has the CHO group. And where the H is placed and OH is placed is very important. So that's how we need to understand the different. See, in Fischer, this is a Fischer projection. The easiest way to understand this is a Fischer projection. The Fischer projection will represent where the OH is placed. Here in the actual D-glyceraldehyde, the OH will be present in the right side. So here none of the component has OH in the right side. So this B is the right answer. 5 of the 6 carbon atom in a dash molecule. So 5 of 6 carbon atom, which means 5 carbon of 6 carbon atom. First of all, the atom is actually 6 carbon. So here only we should consider glucose and fructose is a 6 carbon. So we should immediately cancel out these two options. So 5 of the 6 carbon atoms in a molecule are typically bonded with oxygen. So 5 carbon bonded with oxygen makes a 6 carbon ring. And what is that ring? We have already seen it is a pyranose ring. So what is a component? It is a glucose. If it is a 4 carbon ring with an oxygen, it is a furanose ring and it is a fructose. So we have already seen this. So the answer is actually A. Answer is A, glucose. Okay. So next question. So alpha D-glucose and beta D-glucose are what? It is an anomer. And how do we say this is anomer? Because the anomeric carbon, which is the first carbon, which has an asymmetric molecule, H and OH. So because of the asymmetry, it will produce two types of molecule, which is alpha and beta. So this alpha and beta glucose are ultimately called as anomers. So the option is B. So the next question. Which of the following conformations are more stable? Planar, boat, twisted boat and chain. We have already seen this very well. So the answer is chain conformation. Which of the following compounds are inorganic compound? Okay, this is an interesting question I wanted to include. See, which of the following compounds are inorganic compound? See, most of the people who have studied the food chemistry properly will answer this. If there is an intermediate, like for example, if they are not in touch, surely they won't select water as an option because they thought that water is an organic compound. No, water is actually an inorganic compound because it has no C present in it. There is no carbon in the water, but in fat is present, carbohydrate is present, proteins is present, but in water it is not there. So that's why water is an inorganic compound. So this is a really important question asked in the previous year FSO paper. So make sure to understand the concept of organic and inorganic. So that's it. Here we are concluding the session.